Neuropilin is a very new, promising biomarker for patients with metastatic colon cancer. Data presented that showed that the expression levels measured by ELISA may be predictive and prognostic. Neuropilin is a very interesting receptor which actually is a co-facilitator to the VEGF receptor 1, 2, and 3. And previous data had suggested that low expression level was prognostic in the VELUR trial. So this trial now confirms the potential prognostic value of neuropilin-1. Recent data showed that patients with low levels of neuropilin-1 benefited from bevacizumab. Now these new data show there may particular benefit from a tyrosine kinase inhibitor against VEGF receptor 2, which is the major co-facilitator of neuropilin-1 receptor. So the data are very exciting and are now the basis for prospective validation clinical trials. Dr. Vimasa showing that patients with high expression level of c-Met and wild-type KRAS benefited from a c-Met inhibition in patients who were progressing under EGF receptor blockage. These data are very exciting, even preliminary, showing the proof of principle that when you combine a c-Met inhibition with EGF receptor inhibitors, you may overcome resistance to EGF receptor inhibitors. This is all based on previous preclinical and clinical data suggesting that c-Met is one of the important mechanisms of resistance to EGF receptor inhibition. I think we are waiting now for the second step. These are beyond 21 patients to validate that in a larger expanded cohort. The first 21 patients, 3 patients had an extended response, one of them actually a complete clinical response, indicating that there is a really belief that c-Met inhibition can significantly enhance EGF receptor inhibition even in resistant patients. So I think we are in an area where we have a rich pool of new agents treating the angiogenic pathway. We have approval of bevacizumab, we have approval of aflibercept, we have approval now of ramucirumab, we have approval of regorafenib, and many new drugs are being now developed to target different players in the angiogenic signaling. One of them is nintedanib, which is now in a phase 3 registration trial. These data all suggest that we have a significant impact when you inhibit successfully the angiogenesis. I think what we are lacking behind is to identify patients who benefit most. We really do not have a biomarker. So the story and identification of neuropilin-1 may be one of the first where we maybe identify patients who benefit most from VEGF receptor 2 tyrosine kinase inhibitors, paving the way for more biomarker-driven clinical trials selecting patients for anti-angiogenic treatments. c-Met is much more complicated because I think the challenges with inhibiting the c-Met pathway is to identify patients who have an activation of c-Met. Usually used immunohistochemistry or even FISH is not able to identify very successfully the activated pathways, which means we may not select the patients correctly who benefit from c-Met inhibition. Data from Dr. Vimasa showed really clearly that in a patient who has developed resistance to EGF receptor inhibitors may benefit from c-Met inhibition because that is part of a potential molecular escape. We need to see much more data to see and be confident that this is something we can move forward in larger clinical trials.